Hi guys, good evening. I am Ilangovan, online axi.com person for teaching zoology. So we have started the molecular basis of inheritance, and in this chapter we just start about the transcription process, the first step in gene expression. So in which we talked about the various components of transcription unit, the promoter, what I talked about, then the structural gene, and also the terminator. So the promoter is the one responsible for initiating the transcription process, and to which only normally the enzyme or a polymerase is to be connected or is to be bound for just initiating the process of transcription. And the structural gene, nothing but a group of genes responsible for doing the process of transcription. The message in the gene has been transcribed in mRNA, that is about the structural gene. The termination or the terminator, what we have the specific sequence of DNA responsible for stopping the process of transcription or terminating the process of transcription. So these are all three simple units. We have the promoter, structural genes, and also the terminator, the sequence of DNA, all being formed just on the same strand. Just the promoter towards the 3' direction, upstream. We have different components, some regulatory sequences like enhances, silencers, insulators, etc. So at this key we talked about the two strands of DNA. Out of the two strands of DNA, one strand is acting as a template strand, also called as actually the strand normally not having the original message, but it is acting as a template. Then only the original message found in the complementary strand of DNA would be transcribed and then translated to form the protein. So accordingly we have two different strands we talked about. The last one we talked about the non-coding strand or antisense strand or template strand. So that one is normally having the polarity of 3' 5' direction. Then only we can get the messenger RNA to be synthesized in 5' 3' direction. Now the second strand, the other strand, which is normally called as actually the coding strand or the sense strand, because it is the one having the actual blueprint for the synthesis of a particular protein. But in this case, it is running not in 3' 5' direction as in the case of template string, but it is normally having the polarity 5' 3'. That is different from that of that is the template strand, because you know that one, the two strands being normally anti-parallel. Now, the sequence of actually the bases in this strand is more or less similar to that of RNA, except that in place of uracil or in place of, for example, uracil in the case of RNA, we have thymine in DNA. That is the only difference. Otherwise, the components are same. Now, also I showed actually the picture about the coding and non-coding strand of DNA in the last class. Now let's go back to, suppose here I given that is the nucleotide basis. So I represented the two strands, one in green color, another one just in violet color. So the green colored one is nothing but a strand having the nucleotide sequence, which is acting as a template strand. So I am starting with the nucleotide sequence, that is A, G, C, T, etc. So it is starting from the 3' just ending with that is 5'. That is a running sequence in the case of template strand, that is starting from 3' and then 5'. So these are all the two strands of DNA. These two strands are complementary to each other. You know the meaning for that one. When you have A in this template strand, in the corresponding position, in the coding strand, we have T, because according to the base pair rule, the Chargaff's rule, A is always forming a pair with T. So that is why we have that is T in that place corresponding to, that is A in the first position. So these are all the strands. Let us assume actually A and B strand. And out of these two strands at times of transcription, the only one strand is actually acting as a template. Now this is a template, and this template strand is complementary to the coding strand. And now you see that one, this template strand transcribes the message in mRNA. This process is called copying of mRNA on a template strand, so that we are getting a sequence. Now the sequences found in mRNA after transcription, we see that one, these are all complementary to the template strand. So A is always forming a pair with U, Suppose you have, that is in the case of RNA, or you let us take the second one, G is always forming a parallel to C. So the base sequence in template strand and the base sequence in mRNA strand are complementary to each other. But if you are comparing this mRNA strand, this one, with that of what is called the coding strand, now these two have the bases or nucleotides similar, except in that, see that one, in place of T, you have B. That's the main difference. So in the second case, you have C, here also C. So the original message that is contained in the coding strand, that is why it's called as a coding strand and sense strand, has been transcribed. That is why the other strand is acting as a template. So there is a similarity between what is called the coding strand and mRNA. Both are similar in having the base sequences, excepting that in place of T, in RNA we have U. So this is one illustration what I represent the sequence of nucleotides running in 5'3' direction in the coding strand and in 3'5' direction in the case of template strand. Now let us start with this transcription unit and the gene. So normally you know that what even by gene, a simple mechanism anyone can say, but there is no apt definition for this gene. So normally we can say a gene is nothing but the unit of inheritance, because it is the one which carries the information from one generation to another, transmitting the characters. That is why it's called as unit of inheritance, a simple definition. A gene is nothing but unit of inheritance. Where is it located? It is normally located on the DNA, the place of location on the DNA. Then, I mentioned already it is very difficult to define a gene in terms of its nucleotide sequence. We can say simply a gene that is responsible for the transmission of characters, unit of inheritance. Also we can say the nucleoprotein, what you seen already about, the combination of DNA plus the histone protein to form the nucleoprotein, just like that we have. So the nucleosome, what we have, the unit of the chromosome. And even, so though we have actually, it is difficult to define a gene in terms of its nucleotide sequence. Anything which is responsible for the expression of a particular phenotype is considered as a gene. For example, the DNA sequence, the one which is coding for the synthesis of tRNA or RNA molecule, they are also defining the gene. So the sequence of DNA responsible for the expression of any character, any phenotype. Here I mentioned the tRNA or RNA, they are also being synthesized under the influence of what is called the DNA sequence only, and those DNA sequences responsible for the synthesis of tRNA and or RNA are also defining the gene. So Benzer in 1955 proposed the terms cistron, mutant and recon. Cistron, mutant and recon for the gene. So normally he was the first person in 1955 proposed the term cistron, mutant, recon for the gene. Now what do you mean by cistron? So whether it is cistron or mutant or recon, all are nothing but segments of DNA only. So a cistron is nothing but a segment of DNA that is normally coding or specifying one polypeptide chain. A sequence of bases or a sequence of DNA molecule that is normally specifying one polypeptide chain is called a cistron. It is a functional unit specifying or coding for only one polypeptide chain during protein synthesis. That is what is called actually, that is cistron. Now what are the other two units? Mutant. So it is the smallest segment of DNA actually that can be changed because of mutation. So the smallest segment of DNA that has undergone mutation, that has undergone change, that is called as a mutant, a small segment of DNA that can be changed by a process of mutation. So it is normally a very small segment, it is very small as only one complemental nucleotide pair, only one complemental nucleotide pair. So that is actually the gene mutation frequency is very less. You can have 1 in 100 million genes, we have normally the rate of mutation, highly stable molecule of DNA that cannot undergo mutation just easily, but somehow we have mutation occurs, but it is restricted only one complemental nitrogenous pair pairs, one complemental base pair or nucleotide pair. Then recon, again a segment of DNA that is capable of undergoing recombination, a segment of DNA that to a smallest segment of DNA. It can also be as small as, actually, as small as one complementary base pair of nucleotides. So the smallest segment of DNA that undergoes recombination is called record. So this is the short form, actually, the abbreviated form. And similarly, the muton, the segment of DNA that undergoes mutation. Cistron, the functional unit of DNA that is specific or coding for one polypeptide chain. Now, we are talking about this cistronic gene. And under the cistronic gene, just actually what we are talking about, the structural gene only can be expressed as either RNA, which can be translated to form proteins. So there are two different types of structural gene. One is called monocistronic structural gene. Another one, polycistronic structural gene. Monocistronic structural gene and polycistronic structural gene. So what do you mean monocistronic structural gene? One cistron in an organism that specifies only one polypeptide chain. That specifies only one polypeptide chain, or we can say that specifies the synthesis of one polypeptide chain and not the whole enzyme or a protein. So if you're taking protein, in some cases it is being formed of many polypeptide chains. But normally these polypeptide chains are under the control of many cistrons. So a monocistronic structural gene may be a gene that can normally direct the synthesis of only one polypeptide chain and not the entire protein. Or we can say in another words, a monocistronic structural gene, actually here, one promoter directs the synthesis of one structural gene. One promoter directs the synthesis of actually one mRNA from the structural gene or from actually one cistron. So one promoter directs the synthesis of one mRNA and later that can be translated into only one protein. So there is one promoter that directs the synthesis of one mRNA. That mRNA which is being translated to form only one protein or one polypeptide. And that is very common in the case of eukaryotic genes. The example for monocistronic structural gene, just we call eukaryotic genes. So the gene transcribes one mRNA. That mRNA can be translated to form only one protein. It is very common in the case of eukaryotes. Then the second type, polycistronic structural gene. So in the case of this polycistronic structural gene, it has many open reading frames. The open reading frames, that is nothing but actually the frames having the message. Many open reading frames. And each open reading frame is normally translated into a polypeptide chain. But promoter is one. The formation of mRNA is one. But many polypeptides have been produced by a single, actually what is called a promoter, which directs the synthesis of one mRNA. That mRNA can be translated into more than one protein or more than one polypeptides. But the resultant polypeptides have normally more or less related functions. So, a polycistronic structural gene is a one which has many ORFs, that is open reading frames, and each one just normally is translated into what is called only one polypeptide chain. So, in other words, we have one promoter. There is no change with reference to the promoter, both in the case of monocistronic and polycistronic genes. One promoter, again which directs synthesis of only one mRNA. That mRNA has information for the translation of more than one polypeptides. So in both monocistronic and polycistronic structural genes, one promoter, one mRNA synthesis, but in the first case, formation of one polypeptide chain. The second case, formation of more than one polypeptide chain. So these polycistronic structural genes are very common in the case of prokaryotes, just like bacteria. So monocistronic gene we have, that is in the case of particularly eukaryotes, including human beings, and we have polycistronic structural gene very common in the case of bacteria. There are certain exceptions also. For example, in the case of human being, we have a polycistronic, though I am telling that actually the monocistronic gene is very common in the case of eukaryotes. In some eukaryotes, including human beings, we to have polycistronic structural genes. So an example of hemoglobin. The hemoglobin is actually formed of 4 polypeptide chains, alpha, 2 alpha chains, 2 beta chains. So normally each polypeptide chain is directed for the synthesis by just one promoter, one messenger RNA, but one cistron. So for the formation of what is called one hemoglobin molecule, we need 4 cistrons. And these 4 cistrons are actually considered as a structural gene, working under the influence of just actually one promoter, which direct synthesis of one mRNA. In that mRNA, all the 4 cistrons are transcribed, then later translated to form the proteins. Let's see now the examples. So, though I am telling that in the case of eukaryotes, you have a monocystonic gene, we have also exceptions. So, this is a structural arrangement of the hemoglobin molecule. The hemoglobin molecule is a conjugated protein formed of a heme, an iron containing compound, a porphyrin ring, plus globin, the protein part. The protein part normally contains 586 amino acids arranged in 4 polypeptide chains named as alpha chain. Then we have the beta chain. So there are 2 alpha chains and 2 beta chains. There are 2 alpha chains, there are 2 beta chains. The alpha chains normally contain 146 amino acids. The beta chains normally contain 147 amino acids. So each polypeptide chain is under the influence of one cistron. And the gene contains 4 cistrons for the synthesis of actually 4 polypeptide chains. And all the 4 polypeptide chains have related functions. Nothing but you know that one formation of hemoglobin. So in humans, the gene for hemoglobin synthesis is called as polycystonic gene, though we have generally just what is called the monocystonic gene. So alpha 1, alpha 2, beta 1 and beta 2 are the 4 cistrons considered as structural genes for the synthesis of the hemoglobin having 4 polypeptide chains. So this is an exception and eccentric case we can say. Generally in the case of human beings we have monocystonic gene, but this is an example for the best example for polycystonic gene. Then in the case of E. coli bacteria, we have one system we are going to study later, the lac-apron system, the lac-apron model. So in the case of lac-apron model, so for anything for gene expression we need the help of promoter, what I mentioned. And here just I represented what we have, the gene, the total one, the total gene, what is called an operon system. It consists of a promoter, an operator gene, and these two are normally regulating the 3 cistrons, namely lac Z, Y and A. So these are the 3 cistrons. That's why I am taking polycystonic gene. So a structural gene may contain only 1 cistron or 2 cistrons or many cistrons. If it contains more than 1 cistron, then it's called polycystonic structural gene. It's very common in the case of prokaryotes. So here, this is the structural gene having 3 cistrons. One structural gene having 3 cistrons, lac Z, lac Y and lac A. Now they are being just actually transcribed in mRNA. Now this is the mRNA. We have the genes being transcribed, the 3 cistrons being transcribed, and each one is responsible for the synthesis of particular enzyme. Here Z is responsible for the synthesis of enzyme beta galactosidase, and Y for permease, and for A transacetylase. I mentioned already, if you have polycystonic gene, the products, the proteins have related functions. Now all these 3 enzymes are taking part in one function, nothing but actually the metabolism of lactose sugar. That's why it's called as lac-apron. As it is taking part in the metabolism of lactose sugar, this system is called lac-apron system. We'll be getting more information later separately after finishing this transcription and translation process. So anyway, we have a polycystonic gene is a what? It is nothing but a structural gene having more than 1 cistron. So these are all the 3 cistrons. The best example, the lac-apron system. We also have tryptophan apron. There is another one in the case of bacteria, that is E. coli, tryptophan apron. So there you have 7 cistrons. So here only 3 cistrons. There you have 7 cistrons. In the case of hemoglobin, there are 4 cistrons. So like that. In the case of polycystonic gene, the number may vary. It is a diverse in nature. It is not uniform. It is different from one just species to another species, and also regarding the expression of the genes. Now, I already mentioned about this concept split gene or interrupted gene. So it is very common universal nature, particularly in the case of eukaryotes. We are having split genes, not in the case of bacteria prokaryotes, such an arrangement is absent. So in the case of eukaryotes, what we have the monocytonic structural gene. So definitely, the monocytonic structural gene is called as a split gene. I mentioned about this one earlier also. It is also called interrupted gene because it has some base sequences interrupting the original sequences of function. Those are having functional sequence are interrupted by sequences which are non-functional in nature. That's why it's called as interrupted gene. Unlike the bacterium, there you have only just the coding sequences. There you have only the coding sequences. That's why it's called simply a gene, not a split gene. But in our case, if we are taking human being, the gene consists of both coding sequences, that is the wanted sequences, and also non-coding sequences, unwanted things. So that is why it's called interrupted gene or split gene. A split gene is actually universal in the case of eukaryotes. What's the meaning for that one? So it is a gene that contains a segment of DNA, what are called the exons. These are all the coding sequences, you'll see later, which can be expressed as RNA and what is called the protein. That part, the exons are interrupted by sections of DNA normally, but these sections of actually the DNA, what are called the introns, and they are called as a non-coding sequence of DNA. This is one coding sequence of DNA which do not or which cannot be expressed, or called introns. That is why they are also called as junk DNA, unwanted DNA. They are not taking part in the expression of any protein, but present along with the useful one. So even if you are taking human being, nearly 94% of the genes are split genes. That means we have both the useful sequences and unwanted sequences presented together. At times of transcription or after the transcription process, the unwanted sequences have been removed and only the useful sequences joined together. That is why we have the expression of useful proteins with a phenotypic character. If the process is not done properly, I will come as later. If the process, what process? The removal of unwanted sequences and joining the useful sequences, then we have some hereditary diseases. Nearly 50% of the human hereditary diseases are because of this error, because of non-removal of what is called the sequences of no use. So anyway, the coding sequences are called the exons, which can be expressed as RNA and then proteins. And other sequences of DNA or sections of DNA, what are called the introns, they do not actually have the ability to express themselves, or those which cannot be expressed and translated to form proteins are called introns or non-coding or junk DNA. So in terms of just one line statement, what do you mean by exons and introns? Simply we can say the coding sequences, what I mentioned earlier, or we can have the idea, the expressed sequences. So those coding sequences or expressed sequences are called introns. And this is the one which is normally found both in pre-mRNA as well as processed mRNA. So pre-mRNA having both useful and unwanted sequences. The processed mRNA has only the exons. The unwanted sequence has been removed. So anyway, exons are said to be those sequences that appear in mature or active or processed form. So those sequences normally appear in the mature RNA or processed RNA. After the process, what is called processing is called exons. The other sequence, what will be the introns? These are all actually intervening sequences normally present along with the exons, but they do not appear in the mature or active or processed mRNA. So during the process, what we can say that is a processing, and that is we have one process what is called splicing, you'll see later. So during the process of processing, what will happen? These sequences have been removed because of unwanted function. Unwanted function, we can say they are all not used in expressing a particular phenotype or the protein. So anyway, in a split gene, the exons are interrupted by the introns. So the coding sequences, non-coding sequences, or the expressed sequences, so non-expressed sequences. So this is actually the difference between these two. Again, the exons appear in the processed or active or mature mRNA, whereas the introns do not appear in the mature, processed or active mRNA. Now here is an illustration. So I am taking for example the split gene. So this is a split gene. I am taking 2 exons and 1 intron. So there are 2 exons intervened by 1 intron. So during transcription what will happen? Both introns and exons of DNA have been transcribed. See that one? This is actually RNA precursor or pre-RNA or simply pre-mRNA and processed mRNA or precursor, that is RNA, non-active RNA. So also called as transcript, primary transcript. So without any correction, the DNA has been transcribed. That is why it's called primary transcript or precursor RNA. Now, what will happen? So we need only the exons, we don't want this intron. There occurs a process, what is called splicing, RNA splicing. What is RNA splicing? The unwanted sequences or the non-coding sequences, namely the introns, have been removed. And the coding sequences are joined together to form a mature mRNA. Now this is a mature mRNA, though it's not mentioned here. See that one, it contains only exon 1 and exon 2, the coding sequences only. The non-coding sequence, namely the intron, has been removed. That process of removal of non-coding sequences and joining of the coding sequences to form a mature mRNA or an active mRNA or a processed mRNA is called as a splicing process. So it is simply, we can say, a tailoring process, cutting and then joining. So that is what is called RNA splicing. It is very important just in the process of transcription. Otherwise we will get abnormal protein. The same picture but a little advanced. So split gene in the case of eukaryotes, we have, that one, this part, the promoter area. So in a transcription unit we have promoter. Now this is the gene which is to be transcribed, what is called the structural gene. And then we have one terminator. After this one terminator. Just a sequence which stops the process of transcription. Here I have taken just exon 1 intron, just one intron here. Another intron, the yellow spot, another intron. So I am taking just here 3 introns and 4 exons. So during transcription both introns and exons have been transcribed in the case of what is called mRNA transcript. The unprocessed mRNA is called mRNA transcript. Now it is undergoing the process, what is called, we have splicing, capping and then polyadenylation. We'll have it later. So all these events together called the processing of mRNA. One event splicing, the removal of unwanted. Another one capping, something is added to the front end. And third one adenylation process, tailing process we can say. So we have just a splicing process, removal of unwanted actually mRNA segments. Then capping, something is added to the anterior end or the front end, we can say the 5' end of the mRNA. Then the next one, what is called the tailing process, and during which something is added towards the tail end, that is at the 3' free end. We'll see what will be added. So all these events together call us a processing. So after processing has been completed, we have the mature mRNA. Now this is a mature mRNA. The mature mRNA is having a cap. This is what is called 7 mg cap. 7 mg cap. And also we have what is called adenylyl cap. Adenyl cap. Polyadenylin cap. I'll tell you more about this one later. Then the processed one in the center, you have all the 4 exons joined together. So splicing. This is what is called capping. Something is added to the front end. And this is what is called tailing. Something is added to the just a rear end. That is called the tailing process. All are happening during the process of processing. During the process of processing that resulted in a functional mature mRNA that will be translated to form the protein. Suppose if both exons and introns are actually transcribed and translated, we have a defective protein formation that will lead to the formation of a defective what is called the phenotype. Now, Who are the persons normally responsible for the discovery of split genes? Two different people in 1977, they discovered the split gene. So, Richard J. Roberts, and then Philip A. Shaw in 1977. They discovered the split gene independently. So, in human alone, if you take 94% of the human genes are interrupted genes or split genes out of 100%. So, only 6% alone pure genes. That means having only the coding sequences. The remaining 94 contains both coding sequences and also non-coding sequences, the unwanted things. Then we have a number of disorders, the hereditary diseases. So nearly 50% of the human hereditary diseases involve in splicing introns out. That is normally from the interrupted genes. While doing so, some error is taking place, splicing introns out of interrupted gene. That resulted in what is called splicing error. So one such a disorder, what is seen already under the genetic disease, beta thalassemia. So beta thalassemia is the best example, a disorder caused because of the splicing error, when the introns are actually not being just spliced out of the interrupted gene. So one such a disorder. So it should be careful, just actually the body, the gene. So that is why the frequency of mutation is very, very bigger, or even it is only 1 in 100 million genes. That is why we have the phenotype will not be changed, having the same phenotype unless some radiation injuries. Otherwise the gene is most safe. If the genes are not exposed to the mutagenic agents, then definitely we have the stable structure, stable phenotype not being changed till death. If mutation occurs, any mutagenic agent causes a change in the sequence of DNA that resulted in just what is called defective mRNA formation, leading to the formation of what is called defective protein 2. That resulted in some disorders, what we call this one the genetic disorders. Then let's take the process of transcription. First let us take bacteria. Whether it is prokaryotic cell or eukaryotic cell, for the transcription process to occur, we need the help of three RNAs. So they are called the non-genetic RNAs, and those taking part in protein synthesis are called non-genetic RNAs. So we need badly three different types of RNAs for protein synthesis to take place, and they are designated as messenger RNA or mRNA. We already studied. Transfer RNA or tRNA we have studied, and ribosomal RNA. That is why given only simply the abbreviated form. So these are the three different types of RNAs found in both just prokaryotic cell and eukaryotic cell that are needed for the synthesis of proteins in a cell. Then what is the role? So the mRNA provides a template, a model, because the message has been transcribed in that mRNA, and from that mRNA message, another RNA, what is called the tRNA, reads the genetic code. So the message is given in the form of genetic code. We are going to study later about this one. Altogether we have 64 genetic codons. So the tRNA normally, according to the message present in the mRNA, it reads the message, and accordingly it selects amino acid from the cytoplasm and brings amino acids to the site of protein synthesis. So mRNA provides a template, that means it contains a message after it has been transcribed. Then the tRNA reads the message, because mRNA only coming from the nucleus, the tRNA is found in the cytoplasm. So after transcript, the mRNA leads towards the cytoplasm, where the message is read by what is called the tRNA in the form of genetic code, and that accordingly it brings the amino acid, attaches the amino acid to itself and brings to the place of protein synthesis, namely the ribosomes. In the ribosomes we have another RNA, what is called ribosomal RNA. It plays a structural and catalytic role, that is having the enzymatic activity as well as just playing a structural role, making the formation of the ribosome. The ribosome contains many RNA and proteins, that's why I am using the word structural role, making the ribosome the site of protein synthesis, what we call this one the factory of protein synthesis, and also acting as an enzymatic role, that's why it's called catalytic role. So anyway, each one has its own specific function, that we can say there is a division of labor. So the mRNA provides a template, The RNA brings the amino acid and reads the genetic information of the code, and the RNA play a major role. That is what is called the structural as well as catalytic role in protein synthesis during translation process. Now, what is the main difference? In both the cases I mentioned, just we have three different types of RNAs. Now, in the case of bacteria, there is only one RNA polymerase. As it is working under the control of DNA, this is called DNA dependent RNA polymerase. So, there is only a single DNA dependent RNA polymerase. Single RNA polymerase. And that single RNA polymerase is responsible, it is playing a catalytic role in the synthesis of all the three different types of RNA. Namely the rRNA, tRNA, messenger RNA. So all the three messenger RNAs are synthesized by single RNA polymerase enzyme. So it is working under the control of DNA, that's why we are using the DNA dependent RNA polymerase. So that catalyzes the transcription of all the three types, all types of RNA. But in the case of eukaryotic cells, we have three different types of RNA polymerase, named RNA polymerase 1, 2 and 3. But here only one RNA polymerase can direct the synthesis of three different RNAs. There we have three different enzymes, RNA polymerases. Each one is responsible for the synthesis of either mRNA or tRNA or rRNA. Now, what are the steps involved in transcription process? Let us take in the case of prokaryotic cell, namely the bacterial cell. So, in all cases, we need three steps. One, the initiation. The process has to be initiated. Then, increase in length of the mRNA, the one which is to be transcribed. That is called elongation, by adding the nucleotides. The third process, we have to stop what is called the transcription process. Because suppose we want only 30 amino acids. So we need only the genetic information only for the 30 amino acids. Beyond that one we don't want. So we have to stop the process after finishing the transcription. We have to stop the process. We don't want furthermore the transcription process. And so that we need a terminate. So there are three components or three processes. We can say simply initiation process, elongation of the chain and then termination of the chain. So that we can have just what is called a primary transcript, which is later being actually processed. Now, we have the enzyme. I mentioned in the case of bacteria, there is only one polymerase enzyme, namely the RNA polymerase. The first event for initiation, it has to bind with the promoter region. I mentioned already why is it necessary. This RNA polymerase cannot able to recognize the transcription starting site. It cannot have the ability to find out where is the starting site, namely the transcription starting site or TSS. And it is the promoter, I mentioned about under the promoter, which directs this RNA polymerase to the site of transcription, the starting site. So, for that this RNA polymerase has to just actually bind with the promoter. So, that is the initiation. So, RNA polymerase binds to promoter and initiates the process of transcription. But it itself cannot bind with what is called, that is the binding site, namely the promoter. It needs the help of somebody. So the promoter is recognized again. Otherwise the RNA polymerase cannot recognize where is it actually the promoter region. So the RNA polymerase, actually the promoter is recognized by the RNA polymerase. And another factor, an associated factor, what we call this one, the initiation factor, the sigma factor. So it is needed, but it is not useful to recognize a promoter. Some others are needed. Some people direct this what is called RNA polymerase and the associated sigma factor. This sigma factor is an initiation factor that should be needed for RNA polymerase to initiate the process. But for recognizing the promoter, the sigma factor is not useful. We need a guide. The guide is in the form of what is called an activator protein. So, the RNA polymerase and what is called the associated sigma factor have to reach the promoter and they have to bind with the promoter. For reaching the promoter, they need the help of an activator protein. So, this activator protein is normally binding to its own binding site in the DNA, the one which is found nearby. So first actually the activated protein binds to its own binding site on the DNA. Actually the one which is going to transcribe the RNA, the messenger RNA. And that one guides what is called this RNA polymerase and the sigma factor to reach the promoter. Then only the promoter directs this factor, namely the RNA polymerase enzyme, to the site. So all are interdependent. What is happening only after the other. So the RNA polymerase and the sigma factor, they are normally recognizing the promoter. For recognizing the promoter, for just actually the place where is promoter located, that is called recognition. And that is because of what is called the activated protein. And first the activated protein just normally bind to the DNA molecule to its own site. There also it has its own site. So binding to its own DNA binding site and just giving a guide for this RNA polymerase and the sigma factor to reach the promoter. This is what we can say the initiation process. I mentioned also some of the factors, the transcription factors, general transcription factors I mentioned previously. Recall that one also. Now here the process of transcription in bacteria. I mentioned about actually another word, what is called row independent termination. So comes back later. So we'll have it. Because we have row independent termination and also row dependent termination. So if we want to terminate the process, we need the help of somebody. We also don't want the help of somebody. So here something is needed for terminating the process. In some cases something is not needed for terminating the process. One factor by name the row factor. The row factor is responsible for terminating the process in some cases. In some cases the row factor is not necessary. That's why I use the word row independent termination. That is the last process that comes later. Now what is happening? So the process of transcription, let's see first. The second one will go back later. Now this is what is called the RNA polymerase. The triangle represents the RNA polymerase. Now this is the DNA molecule. Now actually what on the strand is acting as a template. See the red strand is acting as a template. It is a template. So it is called as a non-coding strand and the sense strand or what is called the template strand. And this one, the blue, is the coding strand. So it is not taking part in direct transcription process. So this is factor number 1. What is called the RNA polymerase. The triangle with the green color represents the RNA polymerase. So now this is a promoter in the yellow color. Now this polymerase is now bound with the promoter region with the help of what I mentioned, the factor. So it is not actually doing the process on its own. It needs the help of a factor, what is called initiation factor. That factor is called as a sigma factor. So this is a sigma factor, what we have. So along with the sigma factor, it is normally just actually initiating the process of transcription. So now the process being initiated. Now this is the second process, elongation. So elongation is a process, what is happening there? It is nothing but a polymerization process. The nucleotides are added one after another. The help of the enzyme RNA polymerase. So the RNA polymerase is the main enzyme for actually a chain elongation. So during that chain elongation, what will happen? Only a small stretch of, this is a newly formed RNA. You can see nascent RNA. So only a small stretch of RNA alone bound with what is called actually the polymerase enzyme, not the entire RNA. So it is being added. So only it is found at the tip, adding the nucleotides one after another. But the direction of synthesis is always from 5' to 3' direction. The synthesis always occurs from actually in 5' 3' direction, either the replication process or the synthesis of protein or the transcription of RNA, all being taken place only in the same direction of 5' 3'. That is why we need the template of 3' 5', the DNA. So only small stretch of RNA, we see that one only small stretch of RNA is actually attached or bound with the RNA polymerase. So the termination process, I will come as one, this is row independent termination. That means some sequence being formed in the DNA itself, that is called intrinsic termination. There is no external factor is coming for stopping the termination process. A sequence of DNA in that strand, that is what is called the template strand, is acting as a signal to stop the process of what is called mRNA it stops the activity of RNA polymerase. And finally, see that one, the RNA polymerase, that is a sigma factor, and also the newly formed RNA all being released from the template. This is because of the dissociation process, because of what is called endonuclease complex, an enzyme responsible. So, anyway, this is a simple process without the help of anybody. Here the termination occurs. That's why it's called row independent termination, because in other cases the row factor is responsible for protein for the termination process. But in this case, no row factor is needed for the termination process. We'll come back to the notes. I mentioned already about this one, a bacterial promoter with the Tata box. So, you see that one, this is a promoter G, and here we have the transcription starting site, and this is downstream and this is upstream. What I mentioned, this is plus 1 and this is minus 1. This is already explained. Anyway, the promoter contains, we have a Tata box. There are two consensus sequences, that is in the case of that is bacterial promoter. One at minus 10 region, that sequence is called Tata 80. Another consensus sequence normally present at minus 35 region, that is upstream, is nothing but TTGACG. So these are all the two sequences found in the case of bacterial promoter. Now we see that one is RNA polymerase, the large one, just we have the brown colored one is normally called as RNA polymerase. It needs the help of another person, what is called the sigma factor. This is the oval, the blue oval, the light blue oval represents the sigma factor. Both found attached to the promoter are binding to the promoter. Then only the promoter directs the RNA polymerase to reach. This is the starting site. What it represents the starting arrow mark, where you have the plus 1. This is what is called TSS, the starting site. That is transcription starting site, which proceeds towards what is called the downstream, so that the structural genes have been transcribed. This is what is seen already. This is again I want to expose. Now the second event, what I explained, the elongation process. I mentioned already the RNA polymerase that is responsible for the polymerization process, namely the elongation. It utilizes what is called the nucleoside triphosphate. So nucleoside triphosphates are used not only for what is called this transcription and elongation process, but also the same one used in DNA replication process. So there also we have the nucleoside triphosphates have dual functions. One, it acts as a substrate to be added to the chain. At the same time, it provides energy for the process. So for replication process as well as for this transcription or elongation process, under the transcription for elongation process, we have what is called the nucleoside triphosphates acting as both substrate as well as just acting the energy source. So the nucleoside triphosphate, mostly what is called guanosine triphosphate, and that one is acting as a substrate as well as providing energy for the polymerization process. So the elongation is nothing but a polymerization process. Now the enzyme RNA polymerizes in a template dependent fashion. In a template dependent fashion means one of the strand, the DNA, and we have this is what we have the mRNA being synthesized with the help of RNA polymerase. So in a template dependent fashion, that is by using what is called the DNA template, now it polymerizes, adding the nucleotides one after another to form a long chain of polynucleotide in a template dependent fashion, and follows one rule, what we have the rule of complementarity. Suppose we have in the template strand G, we have C. So this is what is called the rule of complementarity. So it follows the rule of complementarity. So we have G in DNA template strand. Now in the corresponding position, the mRNA we have C. And if you have, for example, A in the corresponding position, you have U. This is what is called the rule of complementarity. And those bases found in the mRNA more or less similar to what is called the coding strand of DNA. So I mentioned only a short stretch of RNA being attached to the RNA polymerase. Always the RNA polymerase catalyzes the reaction only in 5' 3' direction. So I mentioned already, either the synthesis of protein during translation or the replication process of DNA, and or just we have the transcription process Here also the message is transcribed. In all three cases we have the synthesis occurs only in 5' 3' direction. It never occurs in the reverse direction of 3' and 5'. Don't forget. Okay. So this is the elongation, nothing but polymerization process performed by the enzyme RNA polymerase and with the help of energy provided by the guanosine triphosphate. Now termination process. So you have, once the polymerase reaches the terminated sequence, the terminated region, so there is a last point, the terminated destination. What will happen? The nascent RNA, the newly formed RNA, as well as what we call enzyme RNA polymerase falls off. They are being removed, they are being cleaved, they are being dissociated. So anyway, once actually the RNA synthesis has been completed, once the RNA polymerase reaches, we can say that is a terminated sequence of DNA, then the process of RNA synthesis stops. That is called simply the termination process. So that both the polymerase enzyme and the newly formed RNA, mRNA, have been relieved or have been dissociated from the terminated DNA. I mentioned already, in prokaryotes there are two methods of termination process, or termination occurs by two ways. In one method by means of intrinsic termination method or row independent termination. Without the factor, this is row. In one termination, row factor is involved, that is why it is called as intrinsic termination. Row factor is not involved. Some factor, some sequences found within the DNA molecule, that's why it is called intrinsic termination. They cause the termination process. So no factor is involved, namely the row factor, a kind of protein. That's why it is called row independent termination. Another termination, just it is taking place with the help of what is called the row factor. That's why it is called dependent termination, row dependent termination. So independent and dependent. Let's see now what is happening in the case of intrinsic or row independent termination. You see that one actually it is with the help of terminated sequence. Once the RNA synthesis is going to be completed, so before for example stopping the activity of RNA polymerase, this region is exposed and it is normally found in DNA, terminated sequence within the DNA that gives signals to the RNA polymerase to stop the process of synthesis. So here it is activated. It is a person who just stops the activity of RNA polymerase. Immediately the transcription process stops and then the formed RNA has normally dissociated from the DNA template and released into the cytoplasm or released into the nucleus. So in the case of bacteria, you know that one, there are no separate compartments for nucleus and cytoplasm. That's why we can say simply the process, the mRNA has been released in the cytoplasm. There are no two compartments. But in the case of eukaryotes, the mRNA are released in the nucleus, released into the nucleus or simply inside the nucleus where processing occurs. After processing only the processed mRNA pass through the nuclear pores reaching the cytoplasm. That's the main difference. We'll compare it later. So we have the signals for RNA polymerase to stop. The sequence is the terminated sequence. I am using the word T, that is nothing but the terminated sequence. It is a palindrome sequence. So what do you mean by palindrome sequence? Suppose you have in one, this is what we have in the case of equal 1, there is one sequence, palindrome sequence. A palindrome sequence is the one that read the same in both the strands, but in opposite direction. See? So in both the strands we have the same sequences. Here it reads in 5' 3' direction and here it reads in 5' 3' in both the cases. But when we are saying the palindrome sequence, a base sequence that read the same in both the strands, here it is in 5' 3' direction. Here you see that one 3' 5' direction it is running. But in both the cases we have the same sequence but in opposite direction. So anyway the terminated sequence, the intrinsic factor is nothing but the terminated sequence, palindrome found in the DNA, and that is responsible for the stoppage of RNA polymerase activity and relieving the RNA polymerase enzyme from the DNA template. This is what is called the dissociation. So the dissociation of RNA polymerase from the DNA template, that is nothing but the relieving of this one, is brought about by simply the DNA sequence, an intrinsic factor found in the DNA itself, but not coming from outside. That is why it's called as row independent termination. Now the next one, row dependent termination. So here actually this process uses a termination factor. That factor is not present within the DNA. It is a protein factor coming from outside. So that factor is called row factor. So the row factor dependent termination involves or utilizes a termination factor simply called as a row factor. That one sigma factor, initiation factor. The row factor is what is called actually a termination factor. And that is the one being utilized by that just actually the process to stop RNA synthesis at a specific site. At a specific site where we don't want further process. Now, what will happen? This protein is normally binds at the site of the nascent RNA. Suppose this is a nascent RNA. This is binding here, not at the place of where you have the cleaving occurs. So it is binding at a site on the mRNA or the nascent mRNA, the newly formed mRNA, and normally moving runs along in this runs along the mRNA and reaching towards what is called the RNA polymerase, where you have the one which is doing the process. So it is attached to, that is what is called the nascent mRNA. It is moving or running along the entered mRNA and reaching what is called the RNA polymerase enzyme, where it is doing its activity. Then it towards the RNA polymerase enzyme. So when this row factor reaches what is called the RNA polymerase, it causes simply the dissociation of RNA polymerase enzyme from the DNA template. It is the one responsible for breaking the bondage between the mRNA that is formed and also the what is called the polymerase activity being stopped, etc. So it is just like what is called the movement of this one towards this what is called the termination region and stopping the activity of RNA polymerase so that the activity comes to an end. Now we have the row factor, the nascent mRNA, as well as the RNA polymerase all being relieved or cleaved from the DNA template. Now this is the previous one what I mentioned, the intrinsic factor is responsible, but here we see that one. Now the promoter, we have the sigma factor with the RNA polymerase. Now this is the nascent RNA that is being synthesized. So if you want to stop this process, now the process is going to an end. Now the terminator sequence is exposed, but it is not taking part in cleaving that what is called the RNA polymerase and the RNA from the DNA template. Terminator is nothing but the sequence. It is being exposed to stop what is called actually the termination process. But the activities performed here is by the row factor. Now the row factor is responsible for relieving this mRNA, this RNA polymerase, this one is the RNA polymerase, and all being just actually relieved. You see that one? So we have the row factor, then RNA polymerase, all being relieved. Now the newly formed RNA now released. So in the case of prokaryote, there is no processing process. It directly taking part in the translation process. So it is being translated to form protein. So this is what we have. So with reference to what is called the termination process in the case of prokaryotes. So this is once again the same one. I don't want to go through this one, what we had already. Okay. So this is the next question. How is the RNA polymerase able to catalyze all the three processes? Is it possible for a single RNA polymerase to catalyze all the three processes of transcription, that is initiation, elongation and termination? Normally, you know that one, the function of RNA polymerase is only elongation process. It cannot have the ability of actually doing the process of initiation as well as what is called termination. It depends on certain factors. It needs the help of other factors. In association with these factors, this enzyme may normally just actually terminate or initiate the process of transcription. So, and further more information, I would like me just to continue in the next class as the time has been concluded. So till then, goodbye. Now class is complete.